Hello everyone, in this lecture we are going to discuss DNA replication. So in molecular biology, DNA replication is a biological process of producing two identical replicates of DNA from the original DNA molecule. DNA replication acts as a basis for biological inheritance. The cell process the distinctive property of individuals, which makes replication of the DNA essential. So each strand of the original DNA molecule then serves as a template for the production of its counterpart, a process referred to as semi-conservative replications. As a result of the semi-conservative replication, the new helix will be composed of the original one strand as well as the new synthesized strand. So cellular proofreading and error-checking mechanism ensure their perfect fidelity for DNA replications. The experiments were a series of experiments conducted in 1942 by Alfred Hashley and Marcella Chase that helps confirm that DNA is genetic material. At the time, many scientists believed that proteins carried the information for inheritance. In the experiments, Hashley and Chase showed that when bacteriophages, which are composed of DNA and protein, infect bacteria, their DNA enters the host bacterial cells, but most of their proteins does not. They use phage virus as work model. Since phosphorus is contained in DNA but not in amino acid, radioactive phosphor-32 was used to enable the DNA contains in the T2 phage. Radioactive sulfur-35 were used to enable the protein sections of the T2 phages. Because sulfur is contained in proteins but not in DNA. So Hashley's experiments concluded that phosphor-32 containing material entered the bacterial cells. He finally proved that DNA is genetic material and got Nobel Prize. The central dogma of molecular biology describes the two-step process, transcription and translation, by which information in genes followed into proteins, DNA, RNA, and proteins. Transcription is the synthesis of an RNA copy of a segment of DNA fragment. RNA is synthesized by the enzyme RNA polymerase. The classic view of the central dogma of the biological states that the code genetic information hardwired into DNA is transcribed into individual transportable cassettes. Composed of messaging RNA, we call them mRNA. So in the translation program, each mRNA cassette contains the program for synthesis of a particular proteins or a small number of proteins. Semi-conservative replication describes the mechanism of DNA replication. In all long cells, it derives its name from the production of two copies of the original DNA molecules, each of which contains one original strand and one newly synthesized strand. Semi-conservative replication would produce two copies that each contained one of the original strand and one new strand. Semi-conservative replication is beneficial to DNA repair. During replication, the new strand of DNA adjusts to the modification made on the template strand. Conservative replication would leave the two original template DNA strands together in a double helix and would produce a copy composed of two new strands containing of the new DNA pairs. Dispersive replication would produce two copies of the DNA, both containing distinct regions of the composed of either both original strand or both new strand. So nitrogen is a major constitutive of the DNA. E. coli were grown for several generations in the medium containing nitrogen 15 isotopes and nitrogen 14 isotope. The DNA of the cells were labeled by different nitrogen isotopes, and finally, they proved that DNA replication is semi-conservative. DNA replication has 4 key steps: start of replication, or call them origin of the replication, and the initiation site, elongation. Direction is so important during elongation process, and their accuracy. The origin of replication is a particular sequence in the genome at which replication is initiated. The replication fork is a structure that forms during DNA replication. It is created by helix, which break the hydrogen bonds holding the two DNA strands together in the helix. After DNA replication initiation, DNA polymer starts to make new strand of DNA. DNA polymerase is an enzyme that synthesizes DNA molecules from nucleotides, the building block of DNA. The enzymes are essential for DNA replication and usually work in pairs to create two identical DNA strands from a single origin DNA molecules. During this process, the DNA polymerase reads the existing DNA strand to create two new strands that match existing ones. The leading strand is synthesized as a single strand from the point of origin towards the opening replication fork. The lagging strand is synthesized discontinuously against overall direction of replication. This strand is made in many short fragments. It is replicated from the replication fork towards the origin. Base pairing rules must be maintained during this process, and the mistake genome mutation may have consequence on daughter cells. Only correcting pairs fit in the polymerase active site. If one nucleotide is included, polymerase uses its proofreading ability to cleave the phosphodiester bond of improper nucleotides. The activity is from 3 to 5 and then adds correct nucleotide and proceed down the chain along in the 5 to 3 directions. A 3 to 5 proofreading exonuclease domain is intrinsic to most DNA polymerase. It allows the enzyme to check each nucleotide during DNA synthesis and exercise mismatches nucleotides in the 3 to 5 directions. The proofreading domain is also enables a polymerase to remove unpaired 3 overhang nucleotides to create blunt ends. After removal of mismatched nucleotide, DNA polymerase will add new one to allow the synthesis continues. Okazaki fragments are short sequences of DNA nucleotides, which are synthesized discontinuously. Okazaki fragments were later linked together by enzyme DNA ligase to create the ligand strand during the DNA replications. The DNA replication is a process in which new copy of DNA is produced from the parent DNA. When two strands of DNA are separate, each strand acts as a template for the formation of the new strand. This process is called DNA replication. The steps of the DNA replication are initiation, chain elongation and terminations. By mimicking DNA replication in vitro, Frederick Sanger and his colleagues developed DNA sequencing technique in 1977 and got the Nobel Prize. And also, he got another Nobel Prize for the development of protein sequencing technique. Sanger sequencing is a method of DNA sequencing based on the selective incorporation of chain terminating deoxide by DNA polymerase during in vitro DNA replications. This method nourished the blooming of molecular biology in 20th century. Here is some reference, and thank you for your attention.